And next one is Luna Krstić. She's working at University of Valladolid in Spain, and she will tell about formulation aspects at the ocular surface.
Okay, hi, good afternoon to everyone. First of all, I have to say that I'm extremely happy that we managed, even in a reduced amount, to be here today in Paris. And I will give a short overview of the project, of the research project I've been working in the last 3 years, which is about the development of new carriers to improve the bioavailability of topic formulations for natural compounds. I don't know how many of you have actually thought that the dry eye disease could be related to common fruits and vegetables that we consume on a daily basis. Indeed, fruits like apples, grapes, berries, even broccoli or wine are rich in two polyphenolic compounds, quercetin and resveratrol. These two polyphenolic compounds have been designated with a wide range of therapeutic effects, and several years ago in EuDES, they have as well been proven to have a potential therapeutic effect in the treatment of dry eye disease. But their broader application in therapy is limited by their poor physicochemical characteristics, such as the low solubility in water or extremely low chemical stability. And of course, we have tried to have a solution for this. And in the past 3 years, I focused my time on the development of 3 possible formulation strategies to solve these issues. And the first one comprised the use of cyclodextrins, which are widely used pharmaceutical excipients. The second one includes the use of nanoparticles made from elastin-like recombinomers, which are a stimuli-responsive material and biocompatible. And the third option was the most innovative one, and combines the use of 2 delivery strategies, which are therapeutic contact lenses in combination with Micelles. Let's now focus on the first delivery strategy, which was developed in collaboration with the University of Eastern Finland. Cyclodextrins are cyclic oligosaccharides, which possess internal hydrophobic cavity, which is able to host hydrophobic or lipophilic drugs. These cyclodextrins are further able to form inclusion complexes with these drugs. As well, potential addition of auxiliary substances that could eventually form ternary complexes has been explored as a strategy, and in our case we have tried to add hyaluronic acid. Since from the beginning we knew that quercetin and resveratrol possess a low chemical stability, we have tried, we assessed the stability effect of the formation of binary complexes with hydroxy propyl beta cyclodextrin, and the formation of ternary complexes with different concentrations of hyaluronic acid, and we have assessed the stability in a control environment at physiological pH. What we can observe from these stability profiles is that the pure compounds possess low chemical stability, which can be observed in the graphs with black line. Quercetin is extremely unstable, and in 2 days we have its complete degradation, while this degradation kinetic for resveratrol it's not that fast. With the green bars, we can observe the stability of the 2 complexes with hydroxypropyl beta cyclodextrin, and we can see that these profiles were ameliorated. and respectively with blue, orange, and red lines, we have the stability profiles in ternary complexes with increasing concentration of hyaluronic acid. And in this case, we have observed an even higher improvement of the stability of the two polyphenolic compounds. That's why we have decided to, for further studies, make a comparison between the binary complexes with the hydropropyl beta-cyclodextrin and with ternary complexes with 0.1% of hyaluronic acid, since it gave the best stabilizing effect. Since as well we knew that the two compounds possess very low aqueous solubility, we of course have assessed their stability, their solubility when binary or ternary complexes have been formed, and we have observed that this part. We have observed that both quercetin and resveratrol have the solubility improvement with increased concentration of cyclodextrin in solution, and this solubility improvement was maintained as well when hyaluronic acid was present in these complexes. Later on, we have explored the size and morphology of these complexes, and for binary complexes, the size distribution was unimodal, and we can see that the both complexes with quercetin and resveratrol had average size of around 3 nanometers. In the case of the ternary complexes, we can observe that bimodal size distribution was observed, and this indicated that not only small complexes like in the previous case were formed, but as well higher aggregates, and that these aggregates probably were formed due to the presence of hyaluronic acid. And this bimodal size distribution was as well confirmed with atomic force microscopy in both cases. So later on, we have tried to assess the biocompatibility of the binary and ternary complexes in two ocular surface lines, one of cornea and second one of the conjunctiva. And we can see that both type of formulations had a really good biocompatibility, and they did not affect the viability of the two cell line in exam. Later on, we have proceeded to assess the potential capacity of the complexes to neutralize reactive oxygen species that are formed intracellularly, and this was done through the employment of a fluorogenic probe. We can observe that almost in all cases this effect was dose dependent, and with the highest concentration we were able to scavenge more than 95% of the reactive oxygen species that were developed intracellularly. So now the next delivery strategy that we have tried were nanoparticles made from elastin-like recombinomers, and this work was done in collaboration with the Smart Devices for Nanomedicine group from the University of Valladolid. So recombinant polypeptides were obtained from genetic engineering processes, and together with the two polyphenolic compounds, they were dissolved in an organic solvent. Afterwards, this solution was placed in a plant containing supercritical carbon dioxide, and which was acting as an anti-solvent, because it was able to remove the organic solvent from the solution, causing saturation with solutes and leading to a nucleization, which further led to the formation of nanoparticles. We have made 3 types of nanoparticles, one containing just quercetin, the other one containing just resveratrol, and the combination of the two polyphenols. size analysis by dynamic light scattering have revealed that the size distribution of the nanoparticles was quite conserved in all the 3 cases, and this was around 50 nanometers. As well, we have seen that the surface charge of all the particles was negative. Later on, we have proceed to see if we are able to achieve some sustained release. And as a release medium, we have used a simulated tear fluid. We can observe that in around 10 hours, we have a release of 40% of the total quercetin present in solution. As well, we can see in the part of resveratrol that this that the release of resveratrol from the nanoparticles was quite faster, and at the same time point, at 10 hours, we have almost 80% of resveratrol that was released from the nanoparticles. As well, these release kinetics were more or less maintained when release studies were performed on nanoparticles containing both polyphenols. As in the previous case, we have tried to assess the biocompatibility of the nanoparticles in a human corneal epithelial cell line, and we have seen that neither of nanoparticles affected the viability of this ocular cell line. As well, we studied their intracellular antioxidant capacity, and as in the previous case, this effect was dose-dependent, and it seems that nanoparticles containing quercetin were the more efficient in neutralizing intracellular ROS. As to end, I don't know how well we can see it. As to end this part, we have tried to assess the permeation through ex vivo porcine cornea, and this was done with the nanoparticles that possessed a double fluorescent staining. The recombinant polypeptide was marked with fluorescein, and then subsequently nanoparticles were made containing Nile red, which is a fluorescent dye that is used as a model for hydrophobic drugs. Later on, porcine corneas were exposed during a period of 5 minutes to nanoparticles containing Nile red and to pure Nile red. The result that we can, I don't know how much we can see actually, that in the green channel, when porcine corneas were exposed to just pure Nile red, the fluorescent intensity at the level of the corneal epithelium was really modest, while in the case when the porcine corneas were exposed to nanoparticles containing Nile red, this fluorescent intensity was of greater extent. I'm sorry for the quality of the image, but on my screen I see it really well. I'm sorry. As well, in the green channel, what we can observe is that there is a really thin green line present at the level of the surface of the cornea, which means that the nanoparticles are maintained on the corneal surface, and what actually penetrates deeper into the corneal layers, it's the payload of the nanoparticles. And as a last strategy, we have tried to develop some contact lenses embedded with Micelles, and this was done in collaboration with the University of Santiago de Compostela. First of all, Micelles, polaric Micelles containing quercetin or resveratrol were prepared. They were their size were assessed, and they were pretty small, around 5 nanometers. And we also see that these Micelles were very efficient in the entrapment of both polyphenols. In parallel as well, hydrogels made from hydroxyethyl methacrylate or hydroxyethyl methacrylate and methacrylic acid were prepared by methods of thermal polymerization. And after these discs were prepared, they were loaded with the previously prepared Micelles solution through a soaking method. What we can see from the loading studies is that the loading of the Micelles solution in the hydrogels was time dependent, and at a certain time point actually this loading starts to decrease. This actually, I think it was a critical part because we have detected some issues upon release studies that need to be solved. And of course, these are two important parameters that needs to be checked when one is developing a formulation that comprises the use of contact lenses. The first is the optical transparency, which means that the discs that are used need to transmit more than 90% of the light in the visible range, which is from 400 to 700 nanometers. And we can see that all four formulations completed with this request. As well, one important factor is the water uptake, since of this depends the penetration of oxygen through to the cornea, and as well the comfort of the patient while wearing the contact lenses. And usually this parameter is from 40 to 80%, and we from the table we can see that all hydrogels had a very good percentage of water uptake, meaning that the potential they will be very comfortable. So, as to summarize, we have tried to develop three different delivery strategies. The first one was comprising the use of cyclodextrins, with which we have managed to have an improved solubility and stability and a very good intracellular antioxidant capacity. And the second one was with the use of nanoparticles made from elastin-like recombinants, where we have achieved a good solubility. They were biocompatible, and we have seen that they were able to penetrate ex vivo tissues. And the third one, which was with the use of contact lenses embedded with Micelles, we have seen that we have improved solubility when Micelles containing polyphenols were prepared. We complied with good water uptake and transmittance, but we had some issues upon the release of the Micelles from the contact lenses. And so these 3 years, of course, they were filled of lab work, but we have also managed to do some other things. We already published one review which deals about ocular delivery of polyphenols, and we have as well 2 more manuscripts in preparation. I have been to 2 research stays, one at the University of Eastern Finland and one at the University of Santiago de Compostela, and it's already the 3rd year that I'm an invited lecturer in the course of biomaterials in ocular therapy, and where I master my teaching with the students. And as well, last but not the least important slide, because all these results are not just the results of my work, but it's a result of a group of people, of a team. Therefore, I would like to thank my supervisors, my lab mates, all the ERSARs, because we went from colleagues becoming friends, and I would like to thank Paz for the extraordinary work in coordination of everything, all the events and activities that we have, and of course you for your attention.
Okay, thank you, Luna. Any questions? There is one question.
Thank you, Luna, for this presentation. I just, for curiosity, I have two things to ask you. About nanoparticles, how do you plan to introduce to the ocular surface which media you can use, or are you plan to also introduce them into contact lenses for drug delivery, because you cannot directly introduce nanoparticles? And do you have any release studies regarding this? Because you said you have release studies problem with the Micelles and trap to lenses, so you might have the same problem. And second question, why you?
Sorry, can I reply to the
Yes.
first one, because I will forget it. So these are basically two separate projects. Regarding the nanoparticles, they are meant to be used as a eye drop, so in solution. So once the nanoparticles are prepared, they are in solution, probably in a buffer, and applied topically to the eye.
Yes, but when you put in a buffer, what is the storage time? Because they already start to release the drug when they are in a solution, and this will be a problem.
Yes, that's a good point, that the stability was not checked.
Oh, okay. And second question, when we check literature, we see that nowadays most of ophthalmologic drugs are introduced in nanoemulsions, especially cationic nanoemulsions. And you said like your nanoparticles has negative charge.
Yes.
And this will affect your mucoadhesive properties. And why you were not interested to study with cationic nanoemulsions as well?
Yeah, this is also a very good point. Well, the recombinant polypeptide was designed in this manner that it bears a negative charge, because in a repeating sequence of the polymer chain, it has a glutamic acid, which of course bears a negative charge. That's why. And I mean, we have seen from the ex vivo permeation that actually in 5 minutes we reach that the payload is released and that it penetrates the tissue. So I think there should not be further problems in this.
Okay, thank you.
Okay, any other questions to Luna? Maybe I ask you about these ELR nanoparticles. Yes. That you saw that the drug release was something like 10 hours, but then how long do they stay on the ocular surface? Because they have to stay long enough in order to release the drug.
Yes, of course. I mean, we have performed the release studies that was like 1 week. That was actually too much, because we know that once the drugs are applied topically to the ocular surface, they stay less than 5 minutes there. That's why we have moved. We have prepared the nanoparticles that were bearing this fluorescent staining in order to see how can they penetrate the ex vivo tissues. And I have tried several time points. I have shown now the results for 5 minutes, but I actually have done a trial even with 2.5 minutes, and I have seen a fluorescent signal inside the corneal epithelium. So even with lower time, they achieved to release the payload inside the tissue.
Okay. Another point that comes to my mind was that you said that you had some problematic issues with the contact lens
yes.
studies. Is that related to the poor stability of these compounds?
Could be. I think that the problem is, okay, the release studies were done at 37 degrees, and we have already seen that quercetin is unstable at 25 degrees. So probably some degradation factors are interfering with this. And as well, we have to bear in mind that these compounds have a very low solubility in water. Therefore, when we have a release medium that is stimulating the tear fluid, which is of course aqueous, probably we have some precipitation of the drug. So let's say that the study has to be ameliorated. I mean, thought about the design of the study, let's say.
Yeah. Okay, Thank you,
Thank
Luna.
you.